Yeah, good afternoon. Right, as Tom said, I'm the curator of fish collection over at the Florida Museum of Natural History, and more importantly, I have been the advisor for Randy for his 2 graduate degrees, so I will introduce him. He got his bachelor's degree in ecology at the University of Georgia in 2008, and then he found his way to the Florida Museum in 2010. Began working in the ornithology collection, the bird collection, as a collections assistant. Same year found his way to the fish collection. I'm not sure what happened in the bird collection, but anyway, he moved over to fishes, and he worked with us in ichthyology for 3 years. And during that time, he began work on a master's degree in zoology and got the degree in 2013. Master's degree in zoology in the UF Department of Biology. And then he left and took a job at the University of Alabama in Tuscaloosa as manager of the fish collection. Decided he wanted a Ph.D., so he came back to the University of Florida and enrolled in the School of Natural Resources and Environment. And he's been working now, I guess, for 4 years on his Ph.D. His research concentrated initially on systematics of fishes, but he slowly, or maybe quickly, I don't know, became more interested on the use of collections data, the use of data in natural history collections in institutions. He wants to know how the data are used by ecologists and other folks, as well as systematists, who are usually the ones you think of using collection data, but he wants to know who uses the data and why. So during his tenure as a PhD student, he's been employed by iDigBio, which I think most of you probably are aware of. And in iDigBio, he's been strongly involved in outreach activities, excuse me. And he's also worked with several scientific societies, again, on activities primarily related to outreach of and use of natural history collections. He has 7 publications, mostly on, as you would guess, systematics of fishes and natural history collections data. And he has a job waiting for him. He's been employed at the University of Michigan in the fish collection as a collections manager, and he starts on March 1st. So we're really hoping he passes his defense on Thursday, which it's looking good, but you know. And anyway, that brings us to Randy's talk, and he has a title up here, so I won't read it. Randy, it's all yours.
Yeah, thank you so much, Larry, and thank you to the members of my committee here and all of you for showing up. Even if you had to for a class, I still appreciate it. You know, I remember my days sitting in your seat, attending all of everybody else's dissertation talks, and being like, "Wow, I don't know how I'm going to get there." So, anyway, so today I'm going to talk to you guys about 4 years of my life, and try to boil it down into like a 40-minute talk. So here we go. I'm going to talk about an interdisciplinary approach to increasing biodiversity collection sustainability using computational and survey methods. And I promise it's not as dry as the title. All right. So first, for those of you in the room who are not as passionate and integrated in the collections community, I'll give you a quick intro into what biodiversity collections are and what they offer. I'll talk about the first content chapter of my dissertation, where I worked to make a protocol for querying fish biodiversity collections in order to kind of match up the data that is in data aggregators and the specimens and the collections that they belong to. And then from that protocol, we conducted an inventory of digitized fish data in the iDigBio data aggregator, and we presented that to the community and developed a resource that the fish community can use to find and locate fish specimens and reconcile them with their collections. And then finally, I'll talk about my final content chapter, which talks about awareness and use of biodiversity collections by fish biologists, and go over some future directions at the end. All right, so most of the people in the room either work in or have been to a museum. Has anybody in this room not been to a museum before, a natural history museum? See, there we go. We've all been. And we've all seen the wonderful and engaging displays that they have for the public and even for us to enjoy, showcasing the wonderful specimens collected from the natural world. However, as a lot of us know, there is a giant backlog and collection of natural history data that lies behind closed doors that's only usually accessed by people that either work in the collections or use them for their research. And so the real challenge is kind of breaking people through, especially researchers, to know that that data exists and it's there for their use. And we're all of us that are associated with collections, we're all field biologists. We're all out collecting specimens. We're out collecting fishes in the field. We're bringing them back. And we've been doing this for hundreds of years. And all of that data is stored and curated and available for use. There's all kinds of data across all different taxonomic groups. You could study any living organism, and there would be some type of collections data that you could use in your research. When you go more specifically to fishes, one of the largest sources of data that we have are fluid preserved specimens. And so that's fishes collected at a place in time and put in a jar together with a label that has their locality information, any other information about who collected them, where they were collected, all that kind of stuff. And that's stored on a shelf with other fishes. And so that data is useful and available. And we love our collections, right? Like I was just saying, like if you spent 5 minutes in collections, you could probably spend the rest of your life because you realize the power and the uniqueness and the beauty and the community that's involved. And it all centers around the different types of data, like I was saying. So we collect tissue data, so you can get tissue samples, sequences, all kinds of things that are related to any phylogenetic studies. Now, more recently, we've been taking media, so doing CT scans, things like that, and archiving these types of data. Skeletal specimens, so you could get a prepared skeletal specimen or disarticulated skeletal specimen of a fish. Clearing and staining, large specimens. We even have at the Florida Museum, we have tanks of 100-pound sharks, that people can come and look at. And live color photos, so in situ, you can go out, see some fishes in the wild, take some photos, and then bring them back to the museum, and you have both live coloration, habitat information, and the specimen to look at. And from any one specimen, you can glean massive amounts of information. And so it runs the gambit. From even one specimen, you can get anything from isotopes to associated parasites to what food that specimen ate, to field notes, to geographic information. And the real cool thing is that there are some things that specimens might offer to biologists in the future that we don't even know about. So we're still collecting and storing specimens in the best way that we can, hoping that in the future somebody will use those specimens in some way that we don't know yet. In fact, this has been so interesting and important to a lot of people that it actually made it into a book by Mike Webster where he talked about the extended specimen, focusing mainly on ornithological specimens, but talking about all of the different projects and outcomes that can come from just even one preserved specimen. And so when you think about what the real cool thing is about collections, there's two main things in my opinion. One of them is the time component. So as a graduate student, I might go out and collect fishes over a period of 4 years. And that seems like an eternity, right? Like some of you are burning to go tomorrow, you got to finish up. But in the scope of time of someone like your advisor, 20 years of their career, your 4-year span of collecting isn't really that great because they've been collecting for 20 plus years, right? Now, when you think about the entire history of collections and people and famous ichthyologists and famous biologists collecting through time, you have a pretty impressive data set to sample from. And that's what I think is really, really amazing, is that we've created sort of a fish time machine, and it's our job to kind of let them out there and get the community to know that there's this data there for them. So that's one really cool thing. The other really cool thing, and I'll say really quickly, is about geography, right? So as a graduate student, say you're really interested in pomacentrids, which damselfishes, and you get funding to go to the Bahamas and collect some damselfishes and do your study. Well, that might be a really great study, and you might get published, and it might be really interesting and all of that. But if you had supplemented that data with data from collections, you would see a much larger picture. So these are the records of damselfishes in museum collections currently, and you could get this entire geographic scope that you could add to your project, and that you would never be able to do during one period of time for your graduate degree. And it's almost like you were able to clone yourself and send yourself all across the world to get the data that you need, but really you only have to use collections. So how are people actually using collections? That's one of the main drivers that kind of focused me in on what I wanted to do for my PhD. And what most people think is that it's mainly just they're used by taxonomists and, you know, some other stuff too, you know. But actually what we're starting to find more and more in the literature is that people are actually using collections for diverse types of projects. And more with citation methods, people are better citing the resources that they're using. And we're finding out that, yeah, people do use collections in more diverse ways outside of taxonomy and systematics. And so that's something that's really fascinating me. I started as a wee lad at the University of Georgia, where I started my first ichthyological collection, and I've basically walked in one and never left since, because it's something that I feel passionate about. I've tried to help my children learn about the power of collections, tried to tell my family about it and everything, and that's just something that I tried to carry forward in my life. And unfortunately, my son just can't get enough of turtles, so maybe we'll, fish maybe someday. But but not everybody else feels that way, right? So we've all seen the articles recently about collections, especially here in the Southeast, losing funding, losing space, having their programs and budgets restricted. And it's actually a real problem to those institutions, especially the ones that are smaller institutions, small regional collections, small university collections, etc. And there seems to be this stigma about collecting specimens, right? You know, if you're in the museum community, you've all encountered at least one or two people saying, "Oh, you guys are just going out there and, you know, murdering all those animals, and, you know, you're killing so many species." And we all heard about the story about the kingfisher, where it was the last kingfisher of this species. They photographed it and then they immediately preserved it. And, you know, there's some nasty comments, and we've all experienced the negative stigma. But the thing is that once people realize that the amount of collecting that we do, so a large fish collection might be 2 million specimens, 2 million individual fishes. Well, in the time it's taken me to tell you guys about natural history collections, probably that many have died just to make cat food. So when you think about the scale of fishes being eliminated off the planet versus being preserved for scientific research, it really is quite a small number, and the gain that you get out of it is really quite great. And that's something that we've been trying to communicate to the public going forward. And even more recently, I'm sure all of you have known about, there's this article that's been really popular lately about this rodent that has the first casualty of climate change. So this species no longer exists on the planet, and yet you can still go to natural history collections, and this I did today, and you can find 41 specimens of this species in collections available for you to still study and ask questions about this species that you can no longer collect. And there's countless examples that are similar to this as well. And so how does this tie into my dissertation? Well, as Larry said, I went into the collections world for a while, realized there was some problems, and realized that I wanted to address them and through the course of my PhD work. And he's like, what? No. And then after talking for a while, we came up with a good plan of action. And so what can we do as a collections community to increase transparency, increase use, and also what can we do to find out what the community needs, what people want from collections, those kinds of things. And so we need to learn to communicate better. We need to explain our craft better, so explain what we're doing, why we're taking specimens, why we're preserving them. We need to accurately and concisely show what's in collections, because that's a problem. People don't know what we have in our behind closed doors, not a lot of people anyway. And then also find out what researchers need, like I said. And so these main 2 points will be what I'm going to tell you guys about for the chapters of my dissertation. So, how do you get lots of collections data? Well, through digitization, which is the process of taking something that's physical and turning it into something that's digital. So either by taking a photo of it, or adding or typing into a database, or taking data that's already in a database and putting it into a larger aggregate of data, like a data aggregator, you can actually access the data anywhere at any time. And so one of the cool parts about collections, and especially through the I Think Bio initiative, is that collections have been starting to be more digitized. And there are four main data aggregators where collections can send their data, basically publish their data so the public or researchers can come and look at it. And that's IDIG-Bio, FishNet2, GBIF, and VertNet. And so since I'm totally unbiased, I picked IDIG-Bio to do my dissertation work on and look at the data that has been published to IDIG-Bio. But the caveat is that it's also one of the largest, if not the largest, source of data from U.S. collections. So it's a good choice. It's not just my biased opinion. But it's a trap because it's not as easy to get the data that you want as you think it is because of the nature of how data are published. So how are we going to accurately and concisely find what is in collections? It sounds like it might be easy, but after a few weeks of trying to do it, I realized quickly that I had no idea what I was doing and that it was going to be very difficult to get the data and very difficult to do the analyses I wanted to do on my own. And so that's when I was airing my grievances, so to speak, and Kevin Love just happened to hear me. He's another IDIG-Bio employee. He's here in the room. Thank you, Kevin. We worked together exhaustively over the first 2 years of my dissertation to kind of develop a plan of action to address this question. And Kevin is really good at using R. He's an R instructor, and he's really good at coding, and he helped me make my dream into a reality, which we'll talk about in a second here. So there are hundreds and hundreds of collections in the United States across all different taxonomic groups. There are somewhere around 38 collections in IDIG-Bio that would be considered fish collections. And so if we could identify these collections in the IDIG-Bio data aggregator, boom, you got an inventory of collections. But the nature of the way IDIG-Bio ingests data is in a record set manner. And what this means is that most of the time, a record set in IDIG-Bio is a collection or an institution says, I want to publish my data so people can look at our data. Boom, one record set tied to one institution. However, there are several instances where an institution might partner with another institution to publish their data together, or several small institutions might have some sort of grant that requires them to publish their data in a data aggregator, so they come together. So you might end up with a not one-to-one ratio of record sets to collections. So when you go and you look for example, an institution code, and the way you can think of an institution code is UF is an institution. That would be an institution code. Or you can look through collection codes, which think of UF fish collection is like a collection code. And you can't always, so if you look for one of those, in some cases you might not get the collection, you might not get the institution from the collection, you might not get a collection from an institution, or vice versa. And so, since that is the nature of how the data is published, we had to develop a set of protocols to get out which specimens and what data go to which collections. And so there are over 100 million record sets, not 100 records, 100 million records or lots. So like if you're a fish biologist, a lot would be a jar across many, many record sets. So how were we going to reconcile which records go with which collections, go with which record sets, and how to find out how many collections are in IDIG-Bio, how many fish collections are in IDIG-Bio, those kind of questions. And what we found is that we could use a common species like a bluegill or a centrarchid, the family Centrarchidae, to find records within the IDIG-Bio data aggregator and find out what record sets they belong to, and then tie back into what collection they go to. And that culminated in a wonderful and mysterious set of protocols and workflows that looks like this. This is 2 years of hard work from Kevin and I, and you're not really supposed to read it because, you know, it might look like John Madden's football notes or a drawing by my 2-year-old, but this is actually a more distilled version of what we did. So we took all of the data that's in IDIG-Bio, we created a set of 8 queries of using the R IDIG-Bio R package. We did two sets of queries at first. We looked for record sets that had either fish or ichthy in their description, and we also looked through records that had family Centrarchidae and then tied that back into the record set or the UUID that were associated with it. And so using that, we were able to tie individual records to their institution code or their collection code, and then assign a standardized code, which like the American Society of Ichthyologists and Herpetologists has standard codes for all collections in the U.S. and actually abroad as well, but we looked at the US ones, and we assigned a code if there was not a code assigned with it. And then we removed non-extant fishes, removed non-US fish collections, and removed fish collections without fishes. So that, believe it or not, there are several collections in IDIC-Bio that have fishes, but would not be categorized as a fish collection because that's not their primary focus. So we tried to remove those. And once we did that, we had a more distilled query that we could run that removed all of the other extraneous information that we did not need. And then we were able to then standardize things like preparation. So preparation is how a fish was preserved. You know, we have skeletals preparations, clear and stained, etc. And so sometimes it's not always standardized, and so we came up with a set to make that more machine readable and more easily communicable to the community. And also, again, we had to remove some non-fish specimen records. And that gave us theoretically what would be an inventory of fish collections from IDIC-Bio, and we vetted this information with collection managers all across the US. We picked, I think, around 10 to basically call on the phone and be like, hey, did these numbers sound right to you? And in most cases, it was right. And if not, it involved us changing some of our parameters a little bit, and then finally ending up on the final query. And we had two main outcomes, one being that the data that people have in their collections versus what they actually publish to data aggregators is a little bit different. So, for example, I said there's around 38 fish collections in IDIC-Bio. Well, there's 65 unique collection codes, and that comes from collections having multiple collections codes to describe several collections that they house. Like, for example, you might have a tissue collection, a skeletal collection, and your in the jar specimens collection. And some of them report them all separately, even though they're all housed in the same institution. And there's other examples too. And so we use that to reconcile some of those things. In addition, there's a lot of missing data. So upwards of 30% of collection records were missing or had incomplete information on their preparations. They didn't have any individual count, so like the number of specimens in the jar, or they had no family information. In addition, there's also what we call unique iterations of metadata. So that's where you have a lot of different ways to describe the same thing. So one of them is preparations. There were 16,000 different ways that people described a way to prepare a fish, even though there's probably only realistically about 10 maybe. And so that's interesting. And so people are either putting unique codes or they're putting gobbledygook that means something to them but doesn't mean something to us, and it makes machine reading that information very, very difficult. And so these are things that we can give back to the collections community and say, hey, when you publish your data, think about doing some other things. In addition, oops, we got a huge data set for millions of fishes spanning hundreds of years in US collections. And the intersection of different types of metadata that come from just the collection information that gets published is really cool. And you can do you can look at, for example, taxonomic ID with locality, and you can get in some information on biogeography of species of interest and so on. And this goes all across the gambit, and anybody that wanted to do some type of computational ecological analysis of fish distributions or fish information through time could do the same type of query and extract the data points that they found interesting. And the points that we found interesting were these, and we used those to basically conduct the basis for our inventory of fish collections. So, some quick take-homes. Due to the nature of how IDIC-Bio ingests data, matching specimens of data is sometimes a little difficult. Data aggregators can provide vast quantities of data to researchers if they take the time to learn how to use them, and that's a big caveat. It's very hard to use them if you need somebody to show you how to do it. The collections community needs to work on publishing standards and best practices. It's possible to extract an inventory of specimen records in a given set of collections using a data aggregator like IDIC-Bio. And the intersection of metadata can provide answers to a diverse suite of scientific questions. All right, so the second chapter, content chapter of my dissertation, lovingly called Fantastic Fishes and Where to Find Them, looked at US fish collections in IDIC-Bio. So using the same protocol, but applying it to an actual concrete research case. Our main two goals were to create a comprehensive and accurate inventory of aggregated fish collections in the United States, and develop a dynamic resource for the fish collections community to share and compare information. And so the long-term implications of doing a programmatic inventory like this are more than just being like, oh, I'm curious of what's in U.S. fish collections. It gives a better community knowledge of collection holdings, so we know what we're having and what we have to share. Communication of collection superlatives, so I have the most of this, therefore my collection is important. Data set, it gives you a data set for exploring fish collection data. There's better engagement of potential stakeholders. Once you can look across and see what's in collections, then you can better know what you might need for your research. The development of methods for collection data visualization. So how can we best show the data that's in fish collections so the most number of people might want to use it? And then highlighting taxa and geographic gaps in collecting, exploring holdings of rare and valuable specimens. So like where are all our extinct, threatened and endangered, important, not just from a scientific perspective, but from a socioeconomic perspective, where are all these fishes housed? And so the last time an inventory of this type was done before we did it was in 1995 by Poss and Collette. And they basically highlighted a lot of the same points that we tried to highlight through my dissertation, was that if we're not training people to properly curate and communicate the importance of collections data, then we're going to be out of luck, And a reduction of number of scientists engaged in collections-based research will probably impede efforts to study and preserve biodiversity. So less people using collections equals less people helping to preserve biodiversity and study biodiversity. And so they looked across 118 collections. They found 63.7 million specimens, and they did that largely by phone. And so when I was gearing up to do this originally, I got out my phone and I was getting ready to start calling some collections, when I realized, it's 2015 when I started, and we've got computers. So maybe there's a way to programmatically do an inventory of fish collections. And it turned out there was, using the wonderful John Madden or 2-year-old Scribble protocol that I was telling you guys about earlier, and the iDigBio R package. We were able to reconcile fish collections, specimens with collections housed in the iDigBio data aggregator. And so what can we do with these data? Well, once we harvested all of the individual information from collections, we compiled all of that information into a dynamic web resource using a Shiny app. And so that you can go right now to fishfindr.net and explore all fish collection data from iDigBio in the United States. There's several different features that this offers. You can look at individual collections, you can compare across collections, you can look at some maps to show the gaps in collecting, which is one of my favorite talking points when I go and talk about future efforts of collections. So here we see that obviously in the United States, we spend a lot of time collecting in the United States, and we spend a decent amount of time collecting in our neighboring continent of South America, and some smattering of other places like Southeast Asia and Australia. But we can see that there are lots of areas of high biodiversity that have low representation in collections, and that's something we can work on going forward. And I know there's a lot of people working in some of these areas to collect and document the life that are in these locations. And we can report the data. So we can look at how many records, we can look at all the different, and so all these codes on the bottom are collection codes that represent codes or collections in the United States. For example, USNM is the Smithsonian, UF is the Florida Museum of Natural History. And we can compare all different types of preparations across all these different collections. We can look at families, like the number of unique families, which in the fish world, that's kind of how we compare diversity. We look across families. That's a good way to communicate the amount of diversity you have in your collection. And then we can compile all of these data onto individual collection profile pages. So this way collections not only, like every person in their own collection knows what they have, right? Like there's a lot of collection managers in here. You guys know what you have in your collection. But how many of you know what other people see when they look at your collection digitally? Not many people. And so the good part about FishFinder is you're seeing what your data looks like to other people who are searching for your data digitally. And so that gives you kind of a progress report on what's my data looking like once I send it out to be published, how do I compare to other collections, what are our superlatives, how can we work together to better improve our collection, and what sets us apart from other collections. And we even added a compare function to do that. And so you can look at, for example, Oregon State versus UF, and you can have a sweet collection manager duel to see who's got the most. No, actually, you can work together to work on grants and come up with unified efforts to direct the future endeavors of your collection. And that's the idea behind doing that. So, take-homes from our inventory. Since I wanted to spare you guys from going over the nearly 30 graphs that I generated for collections information, just so you know, the top 10 collections hold the majority of all specimens in the U.S. So large collections have a lot of specimens, but there's this trailing tail of smaller collections that have a lot of data as well. Some of the smallest collections have the most diverse preparation types, so even if you're a really small collection, you might have like all of your collection has an image of the live specimen that you collected or something like that. So that means that every collection has something unique and that makes it valuable depending on what you want to look at. Tissue collections still are pretty small. Like not many, there's a lot of museums that aren't even collecting tissues or have very small tissue collections, but they're growing. And also media is becoming more popular, especially CT imaging and 2D imaging being uploaded into data aggregators. And there are many areas of high fish diversity that have low representation of U.S. collections. And finally, large quantities of specimen records doesn't always go proportionally with diversity. So there are plenty of collections I've seen in the U.S. that have lots of specimens, but low number of families. And then there's some collections that have not very many specimens, but have huge representation of diversity. So there's this age-old quantity versus quality that still goes on in collections. So either people are collecting a lot of fishes to get a lot of numbers, or they're collecting, trying to have a large diversity. But all of it's important. All right, so boom, check. Accurately and concisely show what's in collections. Theoretically, we did that. Next, we want to find out what researchers need. And so the title of this chapter that we just submitted for publication is Awareness and Use of Biodiversity Collections by Fish Biologists, aka find out what researchers need. And I presented this at JMIH this year, and we actually did a quiz show where we what did the survey say of what fish biologists, but I figured didn't have enough time to do the whole game show thing, which is really fun. If you want to see it another time, we can do it. Okay, so how can we find out what researchers want? So there's this big thing that goes on within the collections community where people sit around tables, they sit around drinking beer, whatever, talking about who's using our collections. What types of data are the most important? What should we be investing our time in? What should we be investing our resources in? What grant money should go towards different types of collecting efforts? What determines whether or not people want to use collections? And what can be done to increase use of collections in the future? And these are all big questions, and I figured that was something I really wanted to look at. So how did we do it? We did a survey, and we used a Qualtrics digital survey. So via using UF's Qualtrics web applet, I made a Qualtrics survey with the help of Sherry Ellis on my committee. And we disseminated the link. How did we do it? Well, we did it via social media, listservs, personal contact, email, society newsletters, and we estimate that we probably somewhere around 29,000 fish biologists saw the link at least at some point, like they had the option to click on it. And so it got a little overwhelming. Like how are we going to figure out how many people needed to take the survey to have significant results? And luckily there's actually a book about surveys, and it chronicles the number of participants to seeing ratio, and we've calculated that if around 25 to 29,000 people saw it, that we needed about 378 people to take the survey, and we got 432. So my results are significant. Please don't think they aren't. No. So who took the survey? Surprise, surprise. There's a lot of ecologists that are interested in telling us what they think about collections, which was really interesting. And then it ran the gambit. I think we gave them 14 options to pick from, and they could select more than one. And we let them do free response, and I think enough people did physiology, microbiology, and evolution to warrant adding that to the list. So we added those disciplines to the list based on free responses. And in addition, we wanted to look at not just what you're interested in or what you thought was cool, but what are you actually publishing on? And it largely mirrored, what people said they're interested in, but with the small caveat being that there were a decent amount of people from government agencies, private agencies, and things like that, and organizations like that to warrant that about 103 people who took the survey aren't publishing currently. So that's what they do for their job, or that's what they're interested in. So that's who took the survey, but who's actually using collections? And so we boiled down the proportion of individuals who are currently using collections into an individual basis to show the number of individuals who had selected, not just the number of times it was selected. And so ecology was the number one. So people working on ecology are very strongly using collections, which was a very big surprise. And then followed by obviously taxonomy and phylogenetics, which can fall under the same umbrella of systematics depending on how you look at it. But there's a little bit of differences we can talk about later if you're interested. Conservation biology, biogeography, and so on. And then within these different disciplines, we looked at what your professional title was. So it turns out people who would be identified as professional staff are really heavily using collections. And then the more obvious ones like graduate student and faculty members and postdocs are also using collections at a high volume. But there's even some volunteers who are using collections or contract workers. And so what information is the most important? That's the big one, right? We want to know what should we be investing our time and effort into storing. And accurate taxonomic identification across all groups and across all parts of the survey seemed to be the number one resource that collections provide to the research community. They trust that we are going to be putting good, vetted taxonomic identifications from an expert that they can trust. And so that's probably the biggest resource we provide to the research community for free and just readily. But then after that, it was interesting. So a lot of people said that getting meristic or morphometric, so counting number of fin rays or counting scales or things like that, or morphometric, like how long or short a fish was information that they found very valuable. But that is information that we, as collections people, usually just have visitors do when they come. We don't actually provide that information. They do it when they borrow the specimen or visit. So that's kind of interesting. Like if we can find a way to quickly and easily get this data to the researchers, they would find it useful. Like, so if somebody came and took measurements, we could take that data and assign it to the catalog numbers maybe. And then fluid preserved specimens, environmental data. So basically the catch here is that having a physical specimen is extremely important. And anybody that tells you that you can just do whatever you want with a photo and be done with it, with the data, is not even close to encompassing what fish biologists actually need to do their work. They need the physical specimen. So after we asked them what their most useful, we asked them what their top 5 most useful data types were. We looked after that and said, don't pick the same ones again. Now pick 5 more that you find useful. And here's where things got really interesting. So we started to see that there's a huge variance of data that people are using after you start eliminating, taxonomic ID or things like that. We found that field notes are extremely valuable. That's something that collections people keep around. We have field notes all over, but we don't have all of them digitized necessarily. We don't necessarily make them readily available, but that's something that people really want. Another interesting thing that I'll point out, other than the obvious ones like tissue samples and cleared and stained things, which are very project-based. There's people out there that are absolutely nutty about having cleared and stained specimens, but not every collection does that, is early life stages of fishes. And so that's something that a very few collections, at least for fishes, store. But they store them in high quantities. But that's something that obviously people want, so it's something worth investing in the future, especially if you have the infrastructure to maintain a larval fish collection. So my committee really wanted me to break down what data types people found interesting as coordinated to like what their interests were. And so Sherry Ellis on my committee came up with an idea that I really liked, and it's organizing the participants into what we call discipline groups. And so what Sherry did is went through all of the responses. We ended up doing a lot of it together, but she did the coding of it, is look at where the intersections of people's interests are and how they group together. So for example, systematics, neomorphology was a group. So these are systematists who are interested in taxonomy, but they're not looking at morphology, ecology, or behavior. They all did not select those. So they're tied together, and so on. You can look down and see how the rest of them group. And what I find the most interesting is that after Sherry hand-coded it, I went to the UF Stats Consulting to do an actual cluster analysis using their stack system. And Sherry is almost 100% accurate at hand-coding cluster analysis. So I think that deserves a round of applause. And the groups were vetted using the cluster analysis, and we used them to kind of describe if there's any differences between these people who like the same things and the data that they prefer. And actually, when we look at the top 5, we don't really see anything that different. We see, they all kind of want the accurate identification, that kind of stuff, same stuff that you see across the whole group. But what's really interesting is when we look at after the top 5, we get an insane diversity of data types that are wanted across these other groups. And I'm not going to talk about all of them, but if you're interested, I'm glad to go into more detail on what each of these discipline groups found the most interesting. So we also looked at what types of activities lead to future use. No surprise, visiting a collection and getting your hands on doing something in a collection tends to be correlated to people who use it in the future. And if you've never visited a collection or never heard about one, you're almost 100% likely to not even use collections data in your research because you didn't know the resource existed. And then on the flip side, what are the top 3 reasons that people who are not using collections made the decision to not use collections even though they knew about them? And one of them was that the 2 biggest ones were related to awareness and so basically not knowing about resources or not knowing how to access those resources. And that's really key because if we're going to make changes in the community, we have to start giving people better access. And people, like I said, are accessing the data predominantly digitally. So you're downloading data, you're getting data from an outside institution and through digital means. And that's why digitization is still important, and contributing data to these different aggregators is also very important. Another thing I'll go over quickly since I only have a few minutes left is citation. So basically all you need to know is that people either always cite collection resources or almost never cite them. And there's very few people in between. So either you have a good set of protocols for citing what you used, or you just don't care and you don't do it. And my hunch is that the reason we think there's a lot of ecologists that don't use collections is because they fall into the group of people who are not usually citing the resources. And that's just totally speculation. That would be something that would require some more testing. But and then most people are citing the collection code. They're not necessarily, some people are citing the GenBank number for tissues they use, or for sequences they used, and for the data aggregator. Okay, ecologists are a large silent community of users of collections. The most valuable types of collections of data are the actual specimen. They require the actual specimen. Once biologists learn about collections, hey, guess what? They start using them. All types of data are valued by some type of fish biologists, but there are some common data types that are very wanted across all disciplines. Fish biologists want field notes and locality information available online. Early hands-on exposure to collections could be important in future use. Most researchers access collections digitally, so digitization is still important. And adding and curating diverse preparations of data will lead to more use of collections. So we got all this data. So if you are using collections, hopefully these data will help in sustainability efforts, steps towards cohesive data standards, provide a line of feedback from the community on what we as collections people can do to better serve the research community, tools for directing future digitization and curation efforts, and a way to track your place in the community, like I was saying, and what the community sees of your collection. And then if you're not using collections, hopefully there will be a better future for collections in which the data types you want but can't currently get will hopefully be there, and of course for free. That's another caveat, is that all of the data that collections provide is free of charge and you can access open access. Hopefully some better data standards too, to make the data more machine readable. Transparency, so you can see what's in collections and know what's there, and have more awareness of collections resources via better communication from the staff. So a staff member can be like, look at what we got. You can go on to FishFinder, you can go on to IDigBio, and you can see what we have. It's all out there for you. There's no guessing. You don't have to email. You don't have to do anything. So whether you need a time machine or a time capsule, or you need to clone yourself to go across the world to get as much data as you want, you don't need to do any of that. You can just use natural history collections, and hopefully through the work that Larry and I and the committee and Kevin have done over these years, we can hopefully help to make a better sustainable future for collections and the data that they house. Thanks.
Okay, like I said, we have about 5 minutes for questions. Heck?
So, way back in distant time, 1970s, I went to Wisconsin. They couldn't identify fish. The DNR couldn't identify fish. So we set a program up so that we could identify fish. Convinced them that they really should survey the state since it hadn't been done since the 1900s, slightly thereafter. And after we did that, we convinced them it would be nice that they ran surveys. So they did. Surveys were fairly substantial. The material was all collected, but it was divided into 2 parts. One part was the standard collection rules used in systematics and what have you. The other part was preserved in a different manner for destructive analysis. One problem you have routinely is giving up specimens for destructive analysis.
Mm-hmm.
So those were preserved in buffered formula, and they were maintained separately so that you could use them for that. A research project that, for example, in Illinois, looked at the incidence of cancer in fishes. Well, in a century, how would you go about that? Well, they went about it by destructive analysis. They looked at the analysis of cancerous tissue on the digestive system of fishes from the Fox River Valley. Well, some of our collections didn't want to cut their species up. So if we planned a little bit, at least state or local level, to have some destructive analysis material, then that would work. The state of Wisconsin did have it, but then they wiped out their science.
Yeah. And I think that's the goal for collections that keep a large volume of specimens. They do things like that. They try to, that's part of their...
They did end up with U.W. Stevens' point, but they didn't understand the collections that didn't bother doing homework, and they put them in alcohol, which changes the whole ballgame if you're using, if you're looking at lips and things like that.
So when I'm an ecologist.
Yeah.
Right? So I sit there and I think about trying to quantify ecosystem services, right?
Mm-hmm.
And I look at it as like a scientific service.
Yeah.
And so it can be really difficult to quantify, but it's really vital, right, to maintaining and continuing these collection efforts. So I'm thinking when people access data, is there an effort underway to be able to say, hey, before you can use it or download it or whatever, you have to get like an access code or something like that, right? So when you go to publish it, for example, it's kind of like, if you need a permit, a collecting permit or something, and in order to publish that, you have to say, you know what, I use the data from this collection. Do you guys do that?
No, and that's the whole nature of it, is that we want to be more like a library and less like Fisher Scientific. We want basically everything to be open access, open use, and whatever barrier might exist to somebody using it, that tends to be the general consensus, that anything that stops somebody from using collections or hinders them, it's not a good idea. And there are definitely varying opinions with that. Like, I still am of the school where I feel like there should be some type of login, at least to track that you downloaded the data, but it's not always the same for everybody. Yeah.
Because if you can't demonstrate that it's useful, then ultimately the likelihood of you continuing is pretty
It's a huge struggle. Yeah.
We have time for one more.
Yeah, one more.
Supplies over. Okay. Sorry.
How much is known about the origins of collections? Were they usually collected for a specific study, or are there people out there just like, oh, well, we need these, so
It depends how far back you go. If you go back to the real origin of collections, they were basically like a carnival, like a cabinet of curiosities. We collect some things to look at this weird stuff. But there were several collections. Like, in fact, I think Larry, the Illinois Natural History Survey, from where you were before you came to Florida, they were project based and they became a collection. So it's very different depending on where. Yeah.
All right. Randy, really enjoyed that seminar.
Yeah.
Thanks for coming, man.
Thanks for coming, everybody.